My name is Kayla Wilson, and I'm going to be talking about how scientists find an unexpected attraction of nucleic acids and fats, and how scientists found that lipids regulate RNA activity, which can improve the design of RNA vaccines, and it could also explain how RNA could be regulated in synthetic biological systems. This topic stood out to me personally because it has been seen to be a challenge to be able to control RNA activity in the past. There are many things that need to be understood before being able to understand everything that goes with this new finding. First, it would be RNA performs diverse functions ranging from information storage to regulation of other biomolecules and direct catalyst of biochemical reactions. Going through this discovery, it has been demonstrated that RNA lipid binding is influenced by nucleotide content and base pairing of RNAs. Lipids are essential for life, which we all know, and they spontaneously self-assemble to form membranous bilayers, providing a surface that could serve to concentrate, protect, and regulate RNA. RNA can serve the functions of both DNA, which is information storage, and proteins, making it obvious for the need of translation machineries and protein chaperones. Through some effects, RNA-lipid interactions could provide a powerful mechanism for modulating RNA activity. These effects include a mechanism for modulating RNA activity through direct RNA-lipid interactions. Second, alternatively, direct RNA-lipid interactions could localize RNA to membrane surfaces. And lastly, localization to a lipid surface brings RNA into a unique microenvironment. The scientists of this experiment hypothesized basically that RNA membrane interactions could influence ribosome activity by changing the local RNA concentrations. In order to test this hypothesis, there were a couple of different mixtures made to determine the membrane buffer. First, it was a gel phase, and then a ripple phase, and then a liquid disordered phase, and a liquid disordered liquid ordered phase. The RNA lipid buffer indicated how well a particular molecule binded to the membrane. These two scientists discovered that lipids can directly modulate activity of RNA in a simple synthetic system. It is shown that direct RNA-lipid interactions can modulate ribosome activity in a lipid-dependent manner, which means needing assistance of lipids. The major finding was how RNA showed the strongest binding to gel phase membranes and the lowest to liquid disordered membranes. These observations allowed these people to identify that the gel membranes are the most optimal lipid for this study. This chart is just an example of showing how the highest binding was the phospholipid-based gel, which is on the left, and the lowest binding for liquid disorder membranes, which is the one on the right. This image is showing a lipid membrane. The more liquid parts of the membrane are in magenta, and the gel-like parts are in green or dark colored patches. The green RNA molecules focus on the membrane at the gel membrane domains, making the RNA-lipid such a selective interaction. If I was to disagree or agree with the findings of this experiment, I would say I agree, because the scientists left plenty of great charts and information to help me understand each point and how they found out the results to their experiment. Therefore, I would say by their predictions and results, I agree with everything they had to say. The significance of this experiment was to give us more scientific knowledge on how, slowly but surely, an essential framework for engineering RNA lipid systems could be regulated based on sequence, and introduces a mechanism for genetic regulation by RNA in cellular, synthetic, and prebiotic systems.